Hey guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about protein targeting. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. In eukaryotic cells, different proteins need to be sent to different parts of a eukaryotic cell and exported out of the cell into the extracellular space. So our cells have many organelles and all of them require proteins, right? So how they get their required proteins and how extracellular space gets its protein? So proteins must be targeted for these destinations, right? So cells have various shipping systems. In these systems, molecular tags are used to address proteins for delivery to specific locations. This is just like the courier system. When we get our parcels, our address must be well written. Just like that, proteins also have some tags which are used to deliver them in their proper destination. So we will see how they work. So let's talk about the steps of protein targeting. Now, translation of all proteins in a eukaryotic cell begins in the cytosol. After translation, proteins will remain in the cytosol or be transported in the endoplasmic reticulum. So, there are two ways. Either translated proteins will stay in the cytosol or they will be transported in the endoplasmic reticulum. If the proteins have a signal peptide, that means the tag, that is generally an amino acid sequence, they follow the endomembrane system pathway. Endomembrane system consists of endoplasmic reticulum, Golgi apparatus and lysosome. So this is the way: cytosol, ER, Golgi body and lysosome. This is the endomembrane pathway. Now proteins that do not have a signal peptide stay in the cytosol. If they lack other address tags, they will stay in the cytosol permanently. That means they are cytoplasmic proteins. But if they have the other address tags, they can be sent to the mitochondria, chloroplasts, peroxisome or nucleus. Okay. So, what about them whose destination is plasma membrane or cell exterior? We'll see everything. Okay. First, we will talk about the endomembrane system and secretory pathway. Endomembrane system means this pathway: cytosol, ER, Golgi body and lysosome. So first we will talk about it. So proteins destined for any part of the endomembrane system or outside of the cell are brought to the endoplasmic reticulum. How? So the signal peptide that sends a protein into the endoplasmic reticulum is a series of hydrophobic amino acids found near the beginning or end terminus of the protein. So this is the ribosome and here is the protein. This protein needs to be transported in the endoplasmic reticulum. So the end terminus of this protein contains a specific tag, that is the hydrophobic amino acid sequence. Okay, and this is called signal peptide. Now, the signal peptide when emerges from the ribosome is recognized by SRP. SRP is signal recognition particle. SRP brings the ribosome to the ER by binding to a receptor on ER surface. So this is our ER. Here is the ER lumen, this is the ER membrane, and here is the ER receptor. Now this SRP brings the target protein to the ER receptor. Okay. And this receptor binds this peptide sequence. The protein gets inserted into the lumen of the ER, and we can see here the protein is now inserted in the lumen of the ER. Now here is an enzyme. This is signal peptidase. So the enzyme associated with the endoplasmic reticulum receptor, it cleaves the signal peptide. So the signal peptide is now cleaved and the protein is released into the ER lumen. In the ER, proteins fold into their correct shapes and may also get sugar groups attached to them. Most proteins are then transported to the Golgi bodies in vesicles. Some proteins, however, need to stay in the ER and do their jobs there. Actually, ER also requires some proteins. So those proteins will stay in the ER only. These proteins have amino acid tags that ensure they are shipped back to the ER if they escape into the Golgi body. In the Golgi body, proteins may undergo more modifications, such as addition of sugar groups. So some proteins are now transported from ER to Golgi body via these vesicles. And in the Golgi body, they undergo certain modifications such as addition of sugar groups. If they don't have any specific tags, proteins are sent from the Golgi to the cell surface, that is cell plasma membrane, where they are secreted to the cell exterior, just like this. If they are free floating, then they are secreted to the cell exterior, or they are delivered to the plasma membrane if they are membrane embedded. So some proteins will exit Golgi body and they will be transported to the cell surface via transport vesicles. These proteins are of two types. If they are free floating, then they will be secreted to the cell exterior. If they are membrane embedded, they will be delivered to the plasma membrane. Some proteins need to stay in the Golgi itself, since Golgi body also requires some own proteins. So some proteins will stay in the Golgi and do their jobs. These proteins have amino acid tags that ensure they are shipped back to the Golgi if they escape. If proteins are destined for the lysosome, they have a molecular tag consisting of a sugar with a phosphate group attached. So some proteins are very special. They are destined for lysosome. They have a specific tag. That tag contains a sugar and a phosphate group. In the Golgi apparatus, proteins with this tag are sorted into vesicles bound for the lysosome. So we have seen here that how proteins from cytosol are transported to the ER, Golgi body, lysosome, plasma membrane and cell exterior. So this pathway is clear now. Okay. Next we will talk about this non-endomembrane pathway that is targeting to the non-endomembrane organelles. That means now we will talk about those proteins which are destined for mitochondria, chloroplast, peroxisome and nucleus. Let's see. So proteins needed in the peroxisome have a specific sequence of amino acids called a peroxisomal targeting signal. This is a specific signal. This signal consists of 3 amino acids: serine, lysine, leucine, found at the C-terminus end of a protein. This signal is recognized by a helper protein in the cytosol, which brings the protein to the peroxisome. Okay. Mitochondrial, chloroplast and nuclear targeting proteins are generally similar to peroxisomal targeting proteins. That is, a certain specific amino acid sequence sends the protein to its target organelle. However, the nature of the tag is different in each case. So there are many different tags, just like our unique house addresses. So based on these tags, the proteins are separated in different spaces or locations of the cytosol. So this is all about today's lecture. I hope you liked the lecture. Thank you for watching my video.